Picture this. You open your Cambridge Biology 9700 exam paper, and bam, right there is a question on topic 1.2, cell surface membrane structure and function. You smile. You're feeling good, right? I mean, you've basically memorized the textbook. You write down literally everything you know about phospholipids and proteins. But then, results day rolls around, and you find out you lost the mark. No way. Why? Well, it turns out there is a fatal mistake, a trap hidden right in plain sight that costs Cambridge students easy marks every single examination series. So in today's explainer, we're going to expose exactly what that trap is, and way more importantly, how you can avoid it.
Okay, let's dive right into this. If you look at the principal examiner reports, they hit the nail on the head. The absolute #1 way to sabotage your own grade is by providing generic biological facts instead of actually linking your knowledge to the specific membrane scenario they gave you. Look, the examiners aren't just testing your memory here. They're testing your application. Just dropping a memorized paragraph about, say, membrane fluidity without directly answering the specific context of the question, yeah, that is a guaranteed way to lose those crucial grade boundaries. So, let's look at this comparison we have up here. Over on the left, we've got your typical flawed student response. It's kind of generic, it lists some facts, sure, but it totally ignores the experimental context. And, critically, it's completely missing units. Now, check out the top-band answer on the right. It is surgically precise. It takes that concept of membrane permeability and connects it directly to the data provided, complete with the proper units. This right here perfectly illustrates why just doing ordinary rote revision completely fails. Cambridge's assessment framework absolutely prioritizes analytical depth over superficial breadth. You really have to prove that you can think like a scientist, not just act like a walking biological dictionary.
All right, moving into Section 1, Core Principles and the Framework.
If you really want to master this topic, you have to rely on logical reasoning. Think of this as your 3-step insider weapon for absolutely any complex exam problem. Deconstruct, solve, and evaluate. First up, you deconstruct. You do this by identifying the key information given to you in the prompt. Next, you solve it, breaking it down systematically, step by step. And finally, you evaluate. You're checking your answer for reasonableness and making justified recommendations. Top-performing candidates use this exact framework. Having this connected, conceptual understanding is basically what allows them to take their knowledge and easily apply it to those totally unfamiliar contexts that examiners just love to throw at you.
And as you can see, this deconstruct, solve, evaluate framework applies perfectly to the procedural requirements of your practical work and experimental skills. Let's say you're dealing with membrane permeability experiments. You have to systematically select your appropriate apparatus, record your data in suitable tables, spot any anomalies in the results, and then draw conclusions that are based strictly on the evidence. If you try to just skip the framework and jump straight to the conclusion, well, the examiner's absolutely going to dock your points.
Now, in just a minute, I'm going to reveal a massive hidden examiner clue that literally changes your answer completely. It's something most students just entirely overlook. But listen, before I share that with you, you absolutely have to master the data interpretation framework first, because if you don't, this clue is just going to fly right over your head.
So the crucial point is mastering Section 2, the method mark strategy.
Remember, marks aren't just handed out for having the final answer. Cambridge actually heavily awards marks for your statistical process. I know statistics can sound overwhelming, but don't let it stress you out. Just memorize this straightforward framework. If you're comparing the means of 2 different sets of membrane data, you're going to use the t-test. Pretty simple, right? Now, if you're dealing with categorical data, that's when you apply the chi-squared test. And if you're trying to figure out the relationship between 2 variables, like say temperature and membrane permeability, you use correlation tests. Seriously, just knowing which test to use is half the battle won for securing those method marks.
And this brilliantly illustrates the next major leap in what examiners actually expect from you: comparative analysis. Whenever a question asks you to evaluate and compare, examiners are demanding a very, very specific structure. You must identify similarities and differences. You can't just pick one. You also have to use explicit comparative connectives. And above all, you have to support every single comparison with evidence extracted directly from the tables or graphs they give you. If you just point out the differences and move on, you are leaving so many easy marks on the table.
On top of that, doing critical analysis in this topic means you have to actively look for flaws. Really evaluating the quality of evidence means you need to identify any limitations in the experimental setup, you need to note any assumptions that were made along the way, you have to consider alternative interpretations of the membrane data, and then assess how all of this actually affects the final conclusions. To secure that top band, you basically have to act like a peer reviewer who's critiquing a professional scientific paper.
All right, let's move on to Section 3, the Interactive Examiner Challenge.
Okay, it's time to raise the stakes a bit and actually put this framework to the test. I want you to step into the shoes of a Cambridge examiner for a second and look really closely at this real-world data calculation. Go ahead and pause the explainer if you need to, predict, and actively search for the missing elements here. What exactly did this student do wrong? I'll give you a few seconds to spot the errors.
Okay, let's see if you caught it. Looking at the examiner-corrected calculation over on the right, the fatal flaws become pretty obvious, don't they? The student on the left completely forgot to include units in their calculation, and they totally failed to use the correct biological terminology. This isn't just a one-off thing either. The principal examiner reports highlight this over and over and over again. Forgetting units and neglecting precise terminology in your calculations are literally two of the most frequent ways students just bleed easy marks.
Let's move to this next part and see how this all builds into our ultimate checklist. There are 3 massive examiner warnings you absolutely must carry into the exam hole for the cell surface membrane topic. Number 1, watch out for answers that are not specific to the given context. Number 2, do not forget to include units in your calculations. And number 3, failing to double-check your units and terminology during your evaluation phase.
Which leads us perfectly into Section 4, the final exam diagnostic.
Now, if you've mastered all the previous steps, you're well on your way to the top band. But your exam prep is still actually incomplete. Because you could have perfect data, you could have absolutely flawless comparative analysis, and you could still score an absolute 0. Yeah, seriously. Remember that hidden examiner clue I teased earlier? It's the command word. If you mistake an explain question for a describe question, all your beautiful data and precise units mean absolutely nothing to the examiner. Explaining requires scientific reasoning, the why and how. But describing? That just requires you to state what is physically happening. Mixing these two up is the ultimate silent killer in Cambridge biology.
You just cannot afford to lose marks because you misread a single word at the start of a prompt. It's too important. So, to solve this command word crisis, subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCS to excel in your Cambridge examinations. I will see you in the next explainer, where we're going to break down these command words so you can secure your top grade. But until then, I want you to ask yourself, when you look at your next exam paper, are you just describing the biology, or are you actually explaining it?